Okay. Okay, so I'm Kai.
And I'm Annika, and we are presenting senescence reprogramming by TIMP1 deficiency, which promotes prostate cancer metastasis.
And our figures were 3E through H.
Okay, so for the previous figures, 3A through 3E, the premise was that all the mice were treated with docetaxel, and the purpose was to determine whether tumor-induced senescence enhances metastasis or not. The first figure showed a timeline beginning at the treatment at 9 weeks up until 15 to 20 weeks. And then docetaxel treatment did induce senescence in both genotypes of the mice. They used various stains such as SA beta gal, P16, and Ki67, along with a few others to visualize these results in their tissue samples. And then the results showed that the P10 null tumors did in fact increase cell proliferation. However, the P10 and the TIMP1 null tumors had no major increases between the genotypes. And then lastly, tumor-induced senescence increased senescence related genes in both types of the mice. However, there were variations in only certain mice of CDH2 levels for the P10 and TIMP1 null mice.
Okay, so our figures 3E through H talk about the observations of the effects of senescence on prostate cancer in mice. And they determine whether the tumor-induced senescence increases metastasis in P10 null and P10 and TIMP1 null tumors. And the sequencing analysis showed that senescence tumor cells express high levels of the gene BCL2, which is a cell death regulator. And so in our diagrams, the mice were treated with ABT263 or navitoclax and a vehicle, which is a placebo.
And so for the approach, both genotypes of the mice were treated with docetaxel like the previous figures. Then the samples were observed with certain stains. There was a control group that was set up, and this was represented as the vehicle. Then the recorded increase or decrease in senescence-related genes were looked at, as well as CDH2 and metastasis-related genes. And these genotypes were stained with pan-CK and AR for aramine rhodamine and observed in the tissue samples.
Okay, so figure E illustrates the changes in the mRNA of the senescence and metastasis related genes. And so the gray is the control group, and the red group was treated with docetaxel. In figure E, the group was 3 mice, and in F, the groups were 4 and 6 mice. So in senescent cells, the gene BCL2 is increased, which means that they're more sensitive to the docetaxel treatment, since docetaxel treats and targets the BCL2 gene, which is why the red levels are so higher than the gray. And in F, they are PTN null, and in G, they are P10 and TIMP1 null.
Okie doke. So for the following figures, G just represents the stains, as I talked about previously, starting with the docetaxel treatment for both genotypes. In the left column, pan-CK is the type of stain that was used, and on the right is aramine rhodamine. As you can see, in both of the P10 and TIMP1 null samples, there was an increase in metastasis in the mice. And that is also represented in the bar graph on the right, where the right 4 columns of the graph, the P10 and TIMP1 null mice, show that 3 of them did have increase in metastasis, represented by the black bars, versus just the P10 null mice having no metastasis. And the y-axis represents the percentage of the lymph node metastasis that were observed in both 15 weeks and 20 weeks.
All right, so the conclusions for these figures was that tumor-induced senescence, or TIS, shows various results in both P10 and TIMP1 null mice. Both genotypes showed increased levels of senescence-related genes and an increased metastasis-related genes in P10 TIMP1 null mice. The P10 and TIMP1 null mice also increased their percentage of metastasis with the docetaxel treatment. And the P10 null mice had no metastasis in both the vehicle and docetaxel treatment.
All right, and that's it for our presentation. Okay, how do I stop recording it?